Dr. Hansen here again. I hope you enjoyed part 1 on amino acids, and now I want to look carefully at more of the 20 amino acids of life. However, before I go on, I want to make a point. I recommend that you do not listen to this video unless in part 1 you actually tried to draw the structure of alanine, aspartate, and glutamate, and then actually redrew them, replacing the amine group with a ketone. That's so fundamental to the talks I'm going to be giving all year that you will understand my talks a lot better, a lot easier, a lot quicker if you actually do that. If you're going to make a choice between listening to part 2 of the video or drawing those profoundly important amino acids, I would side with drawing the really important amino acids. Assuming, however, you've actually done that exercise, we can go on now. For this video, you're going to be responsible for a number of learning objectives. Some are more important than others, like alanine, aspartate, and glutamate. Next to those 3 amino acids, I've listed about half of the remaining amino acids, which are so fundamental to medicine that I would learn them before I would focus on all 20 structures. I want you also to be able to approximately organize amino acids according to hydrophobicity, and there are some very special amino acids like proline, glycine, cysteine, and methionine, which play very special roles in our life. The good news is that most of you are already kind of familiar with the structure of the 20 amino acids. In fact, a lot of you memorized every one of them a few years ago in introductory biochemistry. If I were you, I would want to take advantage of all that hard work you did a few years ago to memorize all of them. You can refresh your mind now, focus on the most important ones, and review the rest of them. And I think you will then have a much deeper, more complete, and easier understanding of how life works. Before I describe and compare and contrast the various amino acids, I want to begin with a broader perspective and look at polymers of amino acids, or proteins. And of course, protein structure determines function, according to Andrew Taylor Still. Just as the structure of your musculoskeletal system determines its function and affects your health, so the structure of your macromolecules determines their functions. In fact, the structure of protein varies widely, as illustrated by these 2 very diverse types of protein. Can you classify them? Please pause and tell me how you would describe these 2 structures. I would describe the first structure as being globular or globe-shaped. I would describe the second structure of being fibrous. And now I have a question about fibrous proteins. Please pause the video and based on the structure of these 2 proteins, tell me their function. In order to answer the questions, first you need to look at them and say, what do they look like? Structure tells a lot. These fibrous proteins to me look a lot like rope. And what is the function of rope? Rope plays a structural role. It holds things together. And in fact, these 2 fibrous proteins are profoundly important in giving the structure to your body. In health and healing and in neuromuscular, I will talk about collagen. Do you know what fraction of human protein is made of collagen, this structural protein? Please pause and answer. It's about 1/3 of the protein in your body. Why do we have so much collagen in our bodies? Without collagen, the human body would look something like this.
You're officially pardoned from life.
Oh my God!
I'm stepping in it. Look. Wow.
We would essentially be jellyfish, other than the fact that the primary structural protein in jellyfish is also collagen. Collagen is an ancient protein that's widespread through all animal life, going all the way back to the Metazoan radiation. One of the reasons that collagen is so useful to the structure of the human body and to the structure of all animals is its profound modularity which goes through multiple levels of hierarchical structures. For example, it begins with 1,000 amino acid peptide, which is hierarchical to begin with, as is the case of any peptide. You have monomeric amino acid building blocks which get linked into a chain 1,000 amino acids long. In the case of collagen, however, there's an even higher sense of modularity even in the sequence of the collagen peptide itself, because the 1,000 amino acid sequence of collagen is actually comprised out of a 3 amino acid repeat motif, glycine, proline, hydroxyproline, which is repeated over 300 times throughout the 1,000 amino acids of the collagen peptide. So we have a very modular peptide to begin with. Then 3 of these modular peptides wrap around each other to form a hierarchical structure, a triple helix. In turn, this triple helical tropocollagen building block is used in yet a higher hierarchical structure. They're stacked together to make a collagen fibril. In turn, the collagen fibrils are then stacked together themselves to make a hierarchical structure, the collagen fiber, which finally in turn is used in various different orientations and combinations to make even higher hierarchical structures, such as different types of bone, tendons, skin, and other types of extracellular matrix. Collagen is just one example of the profound modularity of life. How very simple building blocks get linked together with simple chemical reactions to make very long, complicated polymers. And this is how simplicity leads to the complexity of life. Let's now move from the fibrous proteins, which give us our structure, to the opposite end of the structural spectrum, to simple spherically shaped proteins, globular proteins. And again, I want you to pause and try to give some explanation of what you think this structural motif may play a role in. And I look at them again, and on a broad perspective, I say, wow, they are spheres. What types of spheres are there in our ordinary everyday life? How about balls? Balls are extremely useful because they're so mobile. You can throw them, kick them, bounce them around. They move very freely. And a number of different types of proteins require freedom of motion. 2 types of proteins which often have to be mobile would be transport proteins and a number of different types of enzymes. The transport protein hemoglobin needs to be in a spherical shape so that it can freely diffuse throughout the red blood cell, bump into oxygen molecules and bind to them in order to transport oxygen throughout the body. Many enzymes, such as lactate dehydrogenase, need to freely diffuse through the cell so that it can freely bump into substrates and catalyze reactions. And specifically, what is that reaction that's catalyzed by lactate dehydrogenase? Please pause and answer. Well, again, you interpret the name dehydrogenase, and it acts upon lactate. So you take lactate, you remove a hydride ion, and this is an enzyme that does it. It turns out that lactate dehydrogenase plays a very important role in linking red blood cell metabolism with liver metabolism, and it turns out that the mechanism by which it removes a hydrogen from lactate is one of the easiest mechanisms of dehydrogenases. Therefore, I want to put this reaction up, and it is the one dehydrogenase reaction I think it's worth your while being sure you understand. Whereas the term dehydrogenase tells you a lot about this enzyme, it doesn't tell you everything. It points you in the direction. But whenever you see dehydrogenase, think, oh, a hydride is going to be driven off of a sugar. Where's it going to go? 9 times out of 10, it's to NAD plus to make NADH. It also may be driven onto FAD to make FADH. There's a couple other compounds. The point is that not all enzyme names tell you everything about the enzyme, but they point in the direction, and in the case of a dehydrogenase, you absolutely have to use a little bit of memorization that's directed by the fact that you know a hydride is being driven off. In particular, lactate dehydrogenase binds to lactate and positions and orients it right next to the nicotine amide ring of NAD+. When the electrons on the hydroxyl resonate down to form a ketone, the lactate gets oxidized to pyruvate. The energy of that oxidation, the pair of electrons, drives the hydride off of the lactate right on to the nearby nicotinamide ring to ruin its aromaticity and make NADH, which has a reduced high-energy hydrogen atom on it. At this point, I would like you to stop the video, and among the two substrates and two products of this reaction, I want you to tell me which is higher in energy, which is lower in energy, which is more oxidized, which is reduced. During the reaction, what got oxidized and what got reduced. Please pause and answer. Okay, a very fundamental principle in chemistry is that when a compound gets oxidized, it goes down in energy. Lactate forms a bond to the electronegative oxygen. It gets oxidized, which means it goes down in energy. NAD plus forms a bond to hydrogen, which is a reduced bond. So NAD plus goes up in energy, such that the overall energy equilibrium between the substrates and products is very similar. This means if there's a lot of lactate, it will tend to form pyruvate, and if there's a lot of pyruvate, it will tend to flow back into lactate. There's also an enzyme called pyruvate dehydrogenase. Can you tell me what that enzyme does? A little bit of a trick question. Pyruvate dehydrogenase does not form lactate. Lactate dehydrogenase, that name implies that the substrate is lactate, and since you take a hydrogen off it, the product is pyruvate. That's for the forward reaction. Lactate dehydrogenase also catalyzes a reverse reaction where the pyruvate actually gets reduced to make lactate. Likewise, pyruvate dehydrogenase is going to be the enzyme that in the forward reaction favors the oxidation of pyruvate to make a more oxidized product. And we will go into this in more detail in the future. It is worth your while to kind of look at the nucleophilic attack mechanism and try to understand what's high in energy, what's low in energy, what goes up in energy, what goes down, what's more oxidized, what's more reduced. Now, I've been looking at structural proteins, and I've been looking at catalysts and transport proteins. I want to point out that proteins are the target of most pharmaceutical agents, and they underlie most genetic diseases. So the better you understand proteins, the better you'll understand medicine. And now I want to take a look at a peptide itself. The peptide backbone, which I'm showing in purple, is identical for the entire length of a peptide. What makes one protein different from another is the sequence of amino acid, which leads to different types of side chains. What is it about the character of a particular amino acid sequence in terms of the different side chains it has that drives the folding of a protein? Please pause and answer. This is something I think you should remember from biochemistry. It is the hydrophobicity of the side chain that drives folding. Accordingly, which amino acids do you think will dominate the folding of this peptide, and roughly how do you think it might fold? The center of folding is especially going to be the large hydrophobic amino acids like phenylalanine and also leucine, which are profoundly greasy and will form the very core of a protein. And as these apolar side chains go from huge to very small, that will determine whether they're completely buried in the core, or in the case of the very small, they're kind of evenly mixed between the core and the surface, because if they're small, their interaction with greasy residues doesn't matter a lot, and their interaction with water doesn't matter a lot. And of course, at the opposite end of the spectrum, amino acids which are extremely hydrophilic are the charged ones, especially like aspartate, glutamate, lysine, arginine. However, before going further with this discussion of how different amino acids contribute to protein folding, I think I need to go in and begin to look more specifically at each of these amino acids. Therefore, I want to return to this list of amino acids. As a medical student, you are not going to be able to learn everything that all of us teach. Instead, you want to decide what is most important and focus first on that, then on what is less important. If you are a biochemistry major, or a chemistry major, or maybe a biology major, you may already be quite familiar with the structure of these very important amino acids, in which case, if you know them cold, it would also be worth your while to focus on what I would consider still important, but less important than the crucial amino acids. You need to pick and choose what will be the highest yield. So again, if you don't know glutamate, glutamine, aspartate, asparagine, alanine, and phenylalanine, along with the related alpha keto acids, then I suggest you go back to part 1. But if you know them, then I would focus next on the important ones and finally on the less important ones. You don't need to know this now, but for the biochemists and maybe some biology majors, 2 of these contain sulfur atoms, and methionine is deeply involved in single methyl group transfer reactions, whereas cysteine is involved in disulfide bonds and in antioxidants. Serine and threonine are important sites of phosphorylation in post-translational modification of proteins. Several of these are quite hydrophobic, and of the hydrophobic ones, 2 of them are beta-branched, isoleucine and valine, and that turns out to be important. Again, depending upon your level of understanding, always focus upon what you already know and then on what's next most important. The order of amino acids in this list is primarily according to medical relevance. But if we organize the amino acids according to structure, then it's easier to learn their structures. So here, I've ordered the amino acids based on increasing hydrophobicity. At the one extreme end, we have the amino acids that have a charged side chain, and charges are almost never buried in a hydrophobic core, so these are almost always on the surface of a protein. Polar amino acids have a hydrogen bonding group, oxygen or nitrogen, but they're not charged, therefore they tend to not be buried also. However, since they are not charged, they sometimes can be buried inside a protein. The hydrophobic amino acids on the bottom tend to be more buried in proteins than the hydrophilic ones on the top, and I've roughly organized the hydrophobic ones from those which are most found on the surface of a protein to those which are most buried at the core. You'll notice I didn't list the names of each of these 20 amino acids, and that's because I want you to have an opportunity to practice. I think right now you should be able to name about half of them based upon recognizing them. In red, I've marked the ones that I think most of you will know and absolutely need to know. And in black, I've indicated ones which also you may not know yet, but boy, it's really in your interest to know. For that matter, go ahead and take a stab even at the ones which I've not put a blank underneath them. You absolutely want to try to label as many of these amino acids as you can. If you get halfway through the list, I think you're in pretty good shape. But go ahead and do that, and then I will put up the names of each of the amino acids, and we will begin to think a little bit about them to help you remember them. Please go ahead, but I need to move on. Here are the labels of each amino acid. Please do confirm that you got all of them right, and hopefully so. And those ones you didn't get right, especially if they're underlined in red or black, do take a look at those and think about them at least a little. Now, before I go on, and look at each of these different groups of amino acids and even at some of the individual amino acids, I want to remind you that the form I've shown here is the form of the free amino acid, which is identical to saying amino acid or identical with saying monomeric amino acids. These are all equivalent words. In fact, it is the form amino acid or monomeric amino acid which floats around in your blood, can be absorbed by cells, and then can be used in protein synthesis or in other forms of biosynthesis. And whereas I talk about charged amino acids, I'm really talking about the side chain, because all amino acids have charges in their free form, in their monomeric form. They have an alpha amine, which is protonated and positively charged, and they have an alpha carboxylic acid, which is deprotonated and negatively charged. Therefore, even if an amino acid side chain is extremely hydrophobic, when you have two charges on it, a zwitterion, then you are very water soluble, even though the net charges cancel to make it neutral. Every one of the 20 free amino acids is remarkably water soluble. However, most of the amino acids in your body right now are actually in the polymeric form called proteins. That's because the positively charged amine of one amino acid has interacted with the negatively charged carboxylic acid of an adjacent amino acid to form a neutral peptide bond. In fact, will you draw the arrows to show the nucleophilic attack of the alpha amine of histidine on the alpha carboxylic acid group of arginine? Please draw those arrows. It's in your interest. If you don't draw the arrows, there's not much point in listening to the video. I simply want you to try to draw the arrows. If it's confusing, you can move on and always draw arrows in future drawings, in future talks. Primary amines are mostly in the protonated positively charged form, but they do exist in equilibrium between a deprotonated neutral form. And the deprotonated neutral form has a lone pair of electrons which can nucleophilically attack the carbonyl carbon in the carboxylic acid. The electrons can resonate up onto the oxygen to form an oxyanion, and then those electrons can resonate back down, and they can actually kick off a water molecule to form a peptide bond. So you'll notice we've taken a positively charged amine and a negatively charged carboxylic acid, and when you form a bond between these two opposite charges, you end up with a neutral bond that's actually quite polar. That means that all of those charged alpha amine groups and all those charged carboxylic acid groups in the backbone of a protein have been completely neutralized, and therefore the only charges that matter are the side chain characteristics. So here I've simply shown all of these linked together, and this is a better way to think about the amino acids, that it's really the side chain that makes the difference in protein structure. In fact, in an entire peptide or in an entire protein, when it comes to the backbone, only two charges remain. Can you identify and name those two charges? Please pause and do so. Well, of course, the two charges that remain is at the N terminus, where there's an N or an amine, and at the C terminus, where there's a carboxylic acid for C. We'll have a positive charge at one end of a peptide and a negative charge at the other. In between, it's all neutral but polar peptide bonds. And it's not critical to know this, but the more correct terminology about an amino acid in a protein is to call it an amino acid residue. And you should be able to identify specific amino acids to know where they begin and end in the structure, because you see the peptide was formed between the carbonyl and the nitrogen, which means that one nitrogen atom, alpha carbon side chain, and the next carbonyl carbon are all part of the same amino acid, and you want to divide them between the peptide bonds, between the carbonyl and the nitrogen, when you're looking at an amino acid as a residue in a protein. Now let's look at the characteristics of the side chains, starting with the positively charged group. Here I've underlined the critical amino acids which I think are most broadly applicable to medicine, in which knowledge of the structure will help you the most. Certainly, those are the ones you should focus on if you're not familiar with amino acids. If you are familiar with amino acids, I'll tell you the more you know about all of them, the better. But again, focus on the critical ones. Let's start with the group of charged amino acid side chains. Aspartate and glutamate. Whenever you see 8, go, oh, 8 is going to be some type of an acid. It is specifically the conjugate base of an acid. Only 2 of the amino acids end with 8, aspartate and glutamate. You absolutely need to know them. And in this case, it's even useful to know that there are 1, 2, 3, 4 carbon atoms in an aspartate amino acid or residue, and 1, 2, 3, 4, 5 carbon atoms in a glutamate residue or amino acid. Of course, when I say this carboxylic acid side chain is negatively charged, I mean it's mostly negatively charged. It's actually in equilibrium between a deprotonated negatively charged form and a protonated neutral form. And that equilibrium will depend upon the pKa value of the acid. As a physician, you absolutely should not know the pKa values of various weak acids and bases. That information is way too specific and useless. What you should know is that given it's an acid, that means it's a proton donor, so that at physiological pH in your blood, carboxylic acids will have a pKa way below 7.4 and will mostly exist in the conjugate base negatively charged deprotonated form. However, given pKa values based on the pH video, you should be able to approximate the amount of conjugate acid, conjugate base for both aspartate and glutamate. Go ahead and do that. It's not the most important part of this video, but you really should be able to work with pH. If you get stuck, do give me a call, text, or email. If we talk about it, you'll find out it's much easier than you think. But I'm going to move on. Given that really detailed, unimportant information that the pKa of aspartate is 3.5, well, the pH of blood is about 7.4, so we're 4 pH units lower than the blood pH. That means that there will be a 10 to the 4 or 10,000-fold ratio. For about every 10,000 deprotonated negatively charged aspartate residues, there will be about 1 protonated neutral aspartic acid residue. Likewise for glutamate with a pKa of about 4.5, that's about 3 pH units below the pH of blood, 7.4, that's a 1,000 to 1 ratio. There will be about 1,000 negatively charged deprotonated glutamate residues in a protein for every 1 protonated neutral glutamic acid residues. In contrast to the negatively charged amino acid side chains, which have carboxylic acids and lose protons to become negatively charged, the positively charged side chains have nitrogen atoms which act as bases and accept protons to become positively charged. It turns out that arginine with its guanidinium group, not a learning objective, and lysine with its primary amine both have picked up a proton and are mostly positively charged in the body. More specifically, I'm going to give you the pKa values again. I'm a little reluctant because I'm afraid some of you might try to memorize them. Please don't. But the pKa value for arginine is about 10.5, and for lysine it's about 9.5. Again, can you give me the ratio protonated, deprotonated, charged, uncharged? But I'm going to go ahead and do that. This is one simple algorithm that I keep asking you to use. Again, the pH of blood is 7.4, which is about 3 pH units below the pKa of arginine. Accordingly, at a pH of 10.5, arginine would be half protonated, half deprotonated, but we're at 7.4, 3 pH units lower. There's a lot more protons. There will be about 1,000
protonated, positively charged guanidinium groups in a protein for every 1 deprotonated neutral guanidinium groups if it's in the blood. Likewise, with lysine residues, there will be about 100 charged protonated lysine residues for every 1 deprotonated neutral one. Hopefully that makes perfect sense. Now you'll notice that histidine, I did not list it with a positive charge, and that's because its pKa is within about a pH unit of the blood's pH. Can you now tell me what the ratio of neutral histidine to protonated charged histidine will be in your blood right now? So 6.3, about 1 pH unit below 7.4. So for every 10 histidine residues, 9 should be deprotonated and neutral, and 1 should be protonated and positively charged. A ratio of 10 to 1 is really quite close compared to all of these other weak acids and weak bases, so it turns out histidine plays important roles in catalysis. In terms of the polar groups, you absolutely should know asparagine and glutamine because of their similarity to aspartate and glutamate and because of the medically relevant interconversion between these forms. Serine and threonine, I don't put on my big list, but they both have a hydroxyl group, they're hydrophilic, and they're often used as sites of phosphorylation. Once you know the more crucial ones, serine and threonine are worth knowing. Cysteine, glycine, and proline are the weirdos. They are special. Glycine, because its side chain is really a simple hydrogen atom. It is so small that other amino acids can freely rotate around it, which makes glycine the most flexible of all amino acids. Likewise, proline is special because proline is the only amino acid that forms a ring that's in the backbone. Histidine, tyrosine, tryptophan, and phenylalanine all have ring structures, but they're outside the backbone and do not completely sterically constrain the backbone and prevent any free rotation. So glycine, the profound outlying flexible amino acid, proline, the extreme outlying rigid non-flexible amino acid. Very important amino acids. Cysteine, importantly, forms disulfides, and it's used catalytically in a lot of reactions. The aromatic amino acids are really interesting too. Phenylalanine by far has the biggest, greasiest side chain, and it is most responsible for protein folding. It is by far the most commonly found amino acid at the deepest part of the hydrophobic core of a protein. It also is used as a precursor to make tyrosine. Whereas tyrosine and tryptophan are both aromatic like phenylalanine, they differ in that each of them has a hydrogen bonding group, and this dramatically decreases how hydrophobic tyrosine and tryptophan are. I've put them near the extreme right side of the figure primarily to keep the aromatic amino acid side chains all together. And in fact, they are still mostly hydrophobic because the single hydrogen bonding group is largely outweighed by the large number of carbon and hydrogen atoms. All of the amino acids on the bottom would be considered hydrophobic. If we look at the very small amino acid side chains, such as alanine and glycine, we see that a single methyl group or a single hydrogen won't form hydrogen bonds to water, so they're not constrained to the surface by hydrogen bonding, nor are they driven from the surface into the core because they do not have many hydrocarbon atoms to interact with. Big greasy chains driven into grease. Very small greasy chains are somewhat soluble. Alanine and glycine are about equally distributed between the surface of a protein and the interior. Just as I have not asked you to focus on serine or threonine, I also am not asking you to focus on valine, isoleucine, leucine, or methionine, because the other amino acids which I've spent so much time talking about in both of these amino acid preview videos are so broadly applicable in medicine. If, however, you've become very familiar with the most important amino acids, next I would tend to look at methionine. because I will be discussing methionine quite a bit, especially with regard to its role in donating its methyl group to cobalamin, vitamin B12, in the cycling of biosynthesis between methionine, cobalamin, and tetrahydrofolate. So it is important, it's just it's not as broadly applicable as the other amino acids which I've been focused on. I only want to go one more step in this talk, and that is I want to look at a hydrolysis of a peptide backbone. How is it that a water molecule could hydrolyze a peptide bond? Please pause and suggest a mechanism, even draw some arrows to show it. Well, of course, we know it's going to be the standard thing, a nucleophilic attack, which means you look for two things. What do you look for? A lone pair of electrons and a double bond. And you're going to say, wait a minute, there's 1, 2, 3, 4, 5. There's a lot of double bonds. And my answer to that is yes. You could attack any one. Let's go for the closest double bond, the carbonyl here. Can you draw the arrows to indicate the movement of electrons? The lone pair of electrons on the water molecule will nucleophilically attack the carbon, which will push one of the pairs of electrons in the double bond up onto the oxygen to form an oxyanion. And of course, those electrons in the oxyanion will be trying to spread out, so they can just resonate right back and reform the double bond and kick off another bond. In particular, as they resonate down to form the double bond, they will push the pair of electrons between the carbon and the nitrogen up onto the nitrogen, breaking the peptide bond while forming an amine, which will pick up protons freely from the solution. Don't care where the protons come, there's a ton of them in water. And you'll notice that the water molecule which hydrolyzes or breaks with water the bond gets added to the peptide, becoming part of the carboxylic acid, which will lose a proton. Again, it is not my intention to teach organic chemistry. However, I absolutely want you as a physician to understand one single type of organic chemistry mechanism, because that will explain 90% of the chemistry going on in your body right now. I think you'll find it more satisfying and easier to learn a lot of medicine if the chemistry is not just a complete black box. Just as formation of peptides in the body are more complicated than simply linking them together, but you need to go through ribosomes and all, so hydrolysis by enzymes involves a little more complexity. But a random hydrolysis is in fact a mechanism by which peptides are broken down. Now I want to look back at some specific amino acids. And I'm going to return to this list of what I really consider to be the key amino acids. This is part of my strategy of trying to address everyone, from someone who has never even had a biochemistry course to someone who's a biochemistry major. If you're not at all familiar with the amino acids, focus on the initial ones: glutamate, glutamine, aspartate, asparagine, alanine, and phenylalanine. That will get you a very long ways. Don't even bother to look at the other amino acids until you really know the initial ones. Once you reach the point of being very familiar with these initial types of amino acids, then go on and look at the others. Or if right now you're stronger in biochemistry and you know quite a few of them, become even more familiar with them. And if you know every amino acid very clearly on this list, I would then go to the amino acids I haven't included, because in fact almost every amino acid at some point will be involved in some disease, and the more you know the better, but you need to start with what you're going to hear the most about and what underlies the most of medicine. Assuming that you've really carefully worked through part 1, I'm going to look at the next most important amino acids, and I'm going to begin by pointing out what I think is important about them. Phenylalanine, tyrosine, and tryptophan are aromatic, greasy, hydrophobic amino acids. Therefore, they are important in driving protein folding. And it turns out that in their monomeric form, they are used as precursors to make neurotransmitters. Accordingly, in neuromuscular,
I will be talking about the biosynthesis and degradation of neurotransmitters built from these aromatic amino acids, as well as diseases that result from genetic defects that prevent biosynthesis of the neurotransmitters or interfere with their degradation, and for that matter, pharmaceuticals that are structurally related to these amino acids and to their corresponding neurotransmitters. Proline and glycine are the extreme outliers when it comes to structure. Proline is totally unique in that it puts a carbon cycle directly in the peptide backbone, completely preventing rotation around one of the bonds. Glycine, with no side chain other than a hydrogen atom, is so small that it completely lets any type of rotation happen around one of its bonds. Lysine and arginine are the two basic amino acids. They can be positively charged. Also, arginine is involved in the urea cycle. Methionine and cysteine both have sulfur atoms in them. Methionine also is used to make S-adenosyl methionine, SAM, and in human development, I'll be talking about single carbon atom transfer reactions, methyltransferases, which require folic acid, cobalamin, and S-adenosyl methionine, along with what happens when those vitamins are deficient. And then finally, cysteine is used in making disulfide bonds. Based on those descriptions, can you identify the amino acids in this peptide backbone, especially proline and glycine? Please look and see if you can identify each amino acid side chain. Good exercise. I'm going to go through it. Well, we have a phenyl ring, so this is a phenyl linked to an alanine, phenylalanine. This ends with a carboxylic acid, so it's glutamate or aspartate, and it has 1, 2, 3, 4, 5 carbon atoms in it between the nitrogen and the carbonyl oxygen, so therefore it is glutamate. How about down here? Again, it has an amide side chain, an oxygen and a nitrogen instead of an oxygen and an oxygen. It has 1, 2, 3, 4, 5 carbon atoms in it. Glutamine. How about this one? It's an 8, so it'll be aspartate or glutamate with 1, 2, 3, 4 carbon atoms. Aspartate. Can you actually see the next amino acid side chain? Where is it? Look here. There's nothing here, but this is an alpha carbon. Here's one peptide bond. Here's the next peptide bond. Just a carbon linking them with 2 hydrogen atoms on it. This is that glycine residue, and you can see how rotating around either bond around the alpha carbon, the whole chain could flip this way, this whole chain could flip this way, because there's nothing to block movement. Let's compare the flexibility of the peptide backbone around a glycine alpha carbon with the flexibility of the peptide backbone around the alpha carbon of a glutamine. In the case of glycine, the entire C-terminus of the protein can rotate around the bond that links the alpha carbon to the carbonyl carbon, because such a rotation is possible because there's no side chain to bump into. Likewise, the entire N-terminus of the protein can rotate freely around the bond that links the alpha carbon to the nitrogen because, again, there's no side chain to bump into. In contrast, if we look at the alpha carbon of the glutamine residue, now if you try to rotate the C-terminus of the protein, there's a good chance that it will bump into the glutamine side chain. Therefore, the rotation is significantly constrained. For the same reason, if we try to rotate the N-terminus of the peptide around the bond that links the glutamine alpha carbon to the nitrogen, likewise it can bump into the glutamine side chain. Whereas most amino acids allow certain rotations to occur, they are constrained when the peptide bumps into the side chain. But with a hydrogen side chain for glycine that's so small that rotations around the alpha carbon are extremely free, and this is conformationally the most flexible backbone alpha carbon.
the guanidinium group, this is arginine. Now here's the big one I wanted you to look for, proline. Do you see how profoundly different that is? Every peptide bond here looks identical, except for the proline, which has a closed ring, which means the bond connecting the alpha carbon to the nitrogen cannot rotate since the side chain links the alpha carbon to that nitrogen. This is an extremely constrained peptide bond. Every other amino acid in this sequence can rotate to some extent. Lysine side chain, histidine, and here's the big difference. You see the histidine? It has a 5-membered ring, just like proline does. And if you look at their monomeric forms, histidine looks kind of like proline. The difference is that the histidine is completely branched off in the side chain, whereas the 5-membered ring in proline is in the backbone itself. Histidine residues can freely rotate around the peptide bond, proline cannot. Really, that's as much of your time as I can take today. I'd like to remind you, don't be afraid to look at these amino acids. One reason I look so much at proline and glycine is that they are extremely prevalent in your proteins. They are the glycine, proline, hydroxyproline sequence motif in collagen that we'll talk a lot about. Again, any questions on any of the concepts from here, I hope you'll contact me. I look forward to your questions, and I look forward to seeing you in Cougar. Thank you.